Hello, I'm Zach Wood. I'm a postdoctoral researcher at the University of Maine, and today I'll talk about some work we've been doing on using environmental DNA to help assess Atlantic salmon in Maine and New Brunswick streams. So first, I need to give a huge shout out to all these government, NGO, and university organizations that have made this work possible. This is huge team science, and without the cooperation of all these different groups, there's no way we would ever have gotten any of this done. So we definitely owe our work to all these folks. And I'd like to thank my co-authors on a couple papers that I'll be highlighting today. A year ago, I hoped this would be brand spanking new data that hadn't been published that I'd be showing you. But because this meeting got delayed a year, now it's all out. It's open access. You feel free to go and look at it, and hopefully you'll find it as interesting as we did. So to give a quick overview on using eDNA for stream organisms, I'm going to use fish, specifically salmon today, but keep in mind you could use this for any stream organism you're interested in. So I see it as there being three sort of hierarchical goals we could use eDNA for here. The first is detection. Are there fish anywhere in this system? Are they there? Well, the second is quantification. How many fish are in the system? And the third is what we're calling quantitative distribution assessment. Just to me, this is sort of the holy grail of eDNA, which would be to say how many fish are in each part of the system. So reach per reach, where are the fish? How many are there? And this is a question we're already trying to answer with electrofishing assessments. And I'm hoping in the future, eDNA will be a tool that we can combine with electrofishing to maybe get some lower cost or higher accuracy with these kind of surveys. But doing this is complicated. So if we have our happy little fish hanging out in a stream with some downstream flow, we've got tissues which break down into cells, which get fragmented, that release eDNA coming off the fish. And we have all these processes like mixing, settlement, denaturation, fragmentation, outwash, that are all complicating the downstream movement of eDNA. So not all sampling locations for eDNA are going to be created equal because of all these processes. Specifically, we have to understand these processes, or at least the general patterns of eDNA dynamics, if we're going to do accurate sampling for fish detection or quantification. So to try to understand these dynamics and patterns of eDNA, we ran a series of cage experiments, first in Maine, then in New Brunswick, again, trying to clarify these downstream dynamics of eDNA with the end goal of helping us come up with good sampling techniques for detecting and quantifying fish using their eDNA. So the first cage experiment we ran, which you can see a nice little picture of here, is in the Sunkhaze stream in Milford, Maine. Beautiful stream if you can put in the effort and survive the mosquitoes to get there. So we put a number of different salmon parr in this cage, and then we measured the eDNA at various downstream distances from the cage. And for this project, we pooled one midstream and two bankside samples, one at each bank, at each sampling distance downstream, which came back to bite us in the butt later, and I think you'll see why. So to give you just a quick rundown of what we saw in this experiment, I'm showing you the quantity of eDNA detected on the y-axis in copies per replicate, and the downstream distance from the fish on the x-axis. And we see this really interesting pattern here, which is that the quantity of eDNA we find is not highest right below the fish. It's highest a little ways downstream from the fish. And you can see I'm pointing out these peaks here. Again, this is 50 to maybe 100 meters downstream from the fish, where we're actually seeing the most eDNA, which is a little mind-boggling and makes us think, what are we missing near the fish? And I think one limitation of this study was that pooling of samples midstream and bankside, and that there might be some sort of dynamic where a lot of our samples close to the fish are actually missing the DNA coming from the fish. So To dig into these dynamics a little bit deeper, we ran a second set of cage experiments in 3 streams in New Brunswick with our Canadian collaborators, with a wide range of salmon par, a wide range of downstream sampling distances, and really importantly, separate midstream and bankside samples to try to tease apart these lateral midstream to bankside eDNA dynamics. And what's cool about this sampling approach is that we get really high resolution ideas of what's going on in these streams. So I'm showing you 9 different stream by fish combinations here. Actually, I think that's 10. I can do eDNA, but I can't count. And so moving from top to bottom, we've got movement downstream from the cage, with the top group here being detection data, the bottom group being concentration data. And the more red a square is, the more DNA we've found. And of course, these are laid out to look like the streams. So you've got a bankside sample and a midstream sample and a bankside sample. So to me, 3 really clear patterns emerge from this graph. The first is that eDNA is most abundant in the midstream near the fish. And that's this red streak coming right down the middle of these graphs here. And then as we move downstream into this 100 to 1,000 meter range, we see with lots of variation that eDNA is roughly well mixed, and that there's sort of this even pattern of color across these regions. And then when we get really far downstream from the fish, so say greater than 1,000 meters, we see that we're actually finding more eDNA near the banks, giving us this sort of split or upside down Y pattern here. And of course, we modeled these patterns. We're not just doing visual analyses, and the modeling also confirms this. And so to show this to you in a bit of a neater way, what we think is happening here is that we're seeing a plume of eDNA. So near the fish, we see this narrow plume with concentrated eDNA that a lot of our samples miss because they're to the left of the right of the plume. As we move downstream, we see lateral mixing of eDNA, bringing it out towards the banks until we've got this sort of wide band of pretty even mixed eDNA midstream and bankside. And then because of the dynamics of the stream, the hydrological dynamics specifically, we get eDNA building up on the banks, so stuck in eddies or falling out onto the stream bottom. And so these models that we built of this plume allow us to ask lots of the interesting conservation questions that I think you're probably here to see. The first being, how far downstream should I sample to detect fish in a specific reach? So we can build up power analyses, which is here just asking how many samples we need to take to detect fish, different downstream distances for bankside samples on the top, midstream samples on the bottom. And our work says that there's a sweet spot, again, roughly from 100 to 1,000 meters downstream, where whether you're midstream and catch the plume or bankside and maybe missing the plume, you're going to have roughly equal chances of detecting the fish and need relatively few samples to do so. Another question is, if you have no idea where the fish might be, how far apart do you want to sample? Say, do you sample every 100 meters, every 200 meters, 300 meters, across the whole stream to just see if fish are there? And again, using the model, we can totally just simulate this and ask that question and answer it. So here we're looking at the detection rate of a few fish just randomly in a stream based on this even sample spacing. And we can get the answer that it's really dependent on like velocity and number of fish, but a good rule of thumb to start with is to sample every 400 meters to detect fish if you don't have any a priori hypotheses of where they are. And lastly, the big question, how do we use eDNA to quantify multiple populations within a stream? And you can take these models that we've built, use some clever sampling designs and some clever simulation math, and you can eventually figure out exactly where and how many fish are in different parts of a stream. And this is complicated, and I don't have enough time to give it the treatment I'd like to, but I can say that if you're looking to do this, regardless of what organization you work for, I'm here to help for free. Please email me, and I'm happy to help you figure out some eDNA sampling designs. So thank you. I look forward to chatting with you later.